Okay, so the last part of this, I know there's a lot of sections, but of videos to watch. You can watch whichever ones you want to based on the things that you need help with. The last part of this is to go over foregut, midgut, hindgut, um, how you create foregut, midgut, hindgut, and basically kind of explain a little bit more why they're so important. And then understanding the difference between continuous or transsegmental structures and non-continuous or segmental structures. We're going to do that hopefully in one last 10-minute video. Okay. So we did all this stuff already. Whoop, done. So now what we're going to do is we're going to look at our embryo in a sagittal section. And I decided to give you guys these pictures and not draw them because I thought maybe it would be harder for you to draw them. So you can use these pictures if you want to, but actually I'm going to delete them right now and draw them myself. Why? Because actually I like to draw it myself. Okay, so we're going to draw a sagittal section of our embryo instead. So that we're going to do the same thing that we've done before. You start at the dorsal hollow nerve tube. Just now, our tube is going to be like that. And this is going to be our head end or our cranial end. And this is going to be our tail or our caudal end. And I'm not going to draw the amniotic cavity over the top. I guess I will. That's the amniotic cavity. That's why I didn't want to draw it. It became like really weird. Then the next thing we're going to draw is, remember, we're going to have the notochord. So I'm going to draw it in black so you can differentiate between a couple of things. And then our next tube that we're going to draw would be our dorsal aorta, like that. And then the next tube that we're going to draw is going to be the gut tube. So the gut tube is going to, actually in very early embryo, is pretty much just going to be a line with like a bubble that comes out like that. So this is our gut tube. So at this point, we still see that basic organization from dorsal to ventral. And I'm just going to label that with some abbreviations: dorsal hollow nerve tube. This is my notochord. This is my dorsal aorta. And then this is my gut tube, okay? Don't use abbreviations on your test. I told you that already, right? So what we're going to see is that we're going to create the gut tube and it rolled up into a tube. We're also going to differentiate or create different partitions of the gut tube after the dorsal hollow nerve tube is already rolled up into a tube. The next thing we're going to have is we're going to have craniocaudal folding or cephalocaudal folding. And essentially what that does is the brain is going to grow super big, super fast. And when it does, it gets really heavy and starts to bend towards the ventral surface. And the tail is also going to start to grow a little bit more and also bend towards the ventral surface. So that creates my brain here and my spinal cord. And to compensate for this bending in the fetal position, to create the fetal position, of course, you know, the notochord is still going to be right here. And why didn't I draw it all the way? Because I kind of have some brain lobes right there. But let's just draw it right there. And we're still going to have the dorsal aorta as well. But because of this head-tail bending, what's going to happen is everything starts to bend towards the ventral side. So everything's going to start to bend towards the ventral midline or towards the middle of the body. And this is going to cause the gut tube to also bend a little bit. So now our gut tube is going to bend right here and it's going to bend right there. And when it does that, you see from the head to the middle part of the embryo, we have like a partition, and then we have like a bubble that comes out like that. So this is essentially going to create foregut structures. So the foregut structures are everything from the pharynx to the first part of the small intestine. and then it's going to create a midgut part. The midgut is going to go from the small intestine to parts of the large intestine. What are those parts right now are a little less important. Just know foregut and midgut. So the head bending creates this little partition or division between the structures of the foregut and the structures of the midgut. As the tail bends, it creates this little partition or division between the hindgut and the midgut. Why is this important? It's important because as you create foregut, midgut, hindgut, what you're going to see is there's going to be a single artery that comes off the dorsal aorta that supplies to the foregut. There's a single artery that comes off to supply to the midgut and a single artery that supplies to the hindgut. So all the structures of foregut have basically one artery that supplies to it. All the structures of midgut have one artery that supply to them. And the hindgut structures have one artery that supply to them. The same thing is going to happen for the veins. It'll have one vein, one vein, one vein. And then what we're also going to see is that coming off of the spinal cord, or really the nervous system, we're going to have one innervation for foregut structures, one innervation for hindgut structures, and one innervation for midgut structures. I should have said midgut and then hindgut. So this means you don't have to try to remember, the artery supply to the stomach. You just have to remember the stomach is part of foregut, and this is the artery supply to foregut. That concept is going to become more important and more apparent in 2240. But for now, you need to know foregut, midgut, and hindgut. The brain bending, the head bending creates the foregut and midgut partition. The tail bending creates the midgut, hindgut partition. So that means when the head and the tail bend at the same time, it creates all three parts, foregut, midgut, hindgut, okay? The other thing we're going to draw here is we're going to start to draw like some segmental structures. So you notice how these tubes run from head to tail. When I drew them, they ran from head to tail, and there's no splitting them up. So just lateral to the notochord, what we're going to see is we're going to start to make those individual blocks that are somites and then intermediate mesoderm. So we're going to draw them maybe right about there so you kind of understand where they are. They probably should be a little bit, let me just redo this. I'm going to make them a little darker color. They're probably about right there on the lateral side, what would probably be the lateral side or the side closer to us, right like that, okay? So somites are individual blocks. That means they're segmental in nature. That means they're not continuous. What this does is if we take sections through our embryo at different parts, when we start to look at this on a chicken or we start to draw our cross sections, and let's say we draw, that's that cross section and that's that cross section. That's why you're always going to see dorsal hollow nerve tube, dorsal hollow nerve tube, notochord, notochord. Notochord up there is black. Dorsal aorta, dorsal aorta. Gut tube, gut tube. Because these are called transsegmental or continuous structures that go all the way from the head to the tail. That's why every cross section is exactly the same. But what you should see is here in this first one, I did cut through my somite and my intermediate mesoderm. So I'll see those in my section. But here I actually cut behind or between the two, so I wouldn't see that. But just so you know, lateral plate mesoderm will also be, remember, continuous or transsegmental. So that would be the difference between these two sections. Here I don't see somites or intermediate mesoderm because I didn't cut through them. Here I do because I did cut through them. If you have a hard time thinking about this or drawing this, you can kind of draw that here, draw your somites here so that you can see they're not continuous. They're interrupted. They're repeating. And remember, that was what my mouse picture was. Each of these little balls is a somite. To help you sort of think about this, you want to think about like chocolate chip bread, right? Chocolate chips are individual, discrete structures. So I have to cut through them in order to see them in my slice of bread. Whereas if you can make really cool, like continuous structures in your bread here, like they do in Japan, maybe you can build yourself a little cat. I think that's a cat. In your bread, every slice that you've taken, you see the same thing. That's called transsegmental. Because it goes the entire length of the bread. The chocolate chips are segmental because they are in the bread, but they're at various places in the bread, so you have to cut them. You have to cut through them in order to see them. Okay, so that's hopefully helpful with what's transsegmental and what's segmental. Things to think about. There's only two things that are segmental. What two things are segmental? I want you to think about that. I want you to write it in your notes because everything else is going to be transsegmental. Okay, think about that. So that means at the end of this video, we divided up our gut tube. So we saw foregut, midgut, and hindgut. And we understand hopefully now that there's like a nerve, an artery, and a vein that are going to specifically supply to all foregut structures, all midgut structures, and all hindgut structures specifically. And I tried to show you what is continuous or transsegmental versus non-continuous or segmental structures. All right. So I would highly recommend you try to print these things out and draw them with me. I might put a little activity up that won't be worth any points, but it kind of keeps you guys on track with the class if you do that and you just submit it as if it's an assignment. I might also make this as a discussion board, so if you have questions, then you can come and ask questions and I'll kind of try to monitor it to answer your questions, and then other students can answer as well.